Assalamu alaikum. As you can see on my YouTube channel, I uploaded a video a few years back that I'm going to start a series of lectures on chromatography, but I couldn't continue to that. So now I decided to continue that series, and today's lecture is part one, you can say, of the chromatographic lectures. So in today's lecture, you will learn what is chromatography, its basic terminologies, and how you are going to perform chromatography, and how you are going to calculate the RF values. So firstly, introduction to chromatography. So chromatography is basically made up of two words, one of which is chromo and the other is graphy. Chromo means coloring and the graphy means representation of something on paper. So basically, with the help of colors, you are representing your different components on a paper, and this paper is basically the stationary phase. And chromatography is a physical method of separation when we talk about the definition, where the components are separated on the base of their affinity for the stationary phase and the mobile phase. So what is chromatography? Chromatography, it is a technique that is used to identify unknown compounds and the separate mixtures. Uh, Basically, it is used for the purpose of identification, to separate the mixture for the purification, and it is also used for analyzing or quantification of the different components present in the mixture or unknown samples. So what is chromatography? Chromatography is a technique for separating mixtures into their components in order to analyze, identify, purify, and quantify the mixture or components. And when I talk about quantification, chromatographic technique itself or with the combination of other techniques can be used for the quantification of the mixture or the components which are desired to be separated from the sample. These are the basic chromatographic terms, one of which is chromatogram. It is a visual output of the chromatograph. Chromatograph is the it enables the sophisticated separation, and stationary phase is the, as I already told you, the paper that is used for the separation or to represent the separated compounds. That is stationary phase. It is the phase that is covalently bonded to support particles or the inside walls of the column tubing. Mobile phase, it is the phase which moves in a definite direction. These are the solvents. Single solvents can be used, or in a combination they can be used in different ratios. Analyte, it is a substance which is to be separated during the chromatography or the desired component which we want to separate. Eluate, it is the mobile phase leaving the column. Retention time, it is a time which is taken by the component to be separated. It is a characteristic time it takes for a particular analyte to pass through the system from the column inlet to the detector under set conditions. Eluent, it is a solvent that will carry the analyte. What are the requirements to perform the chromatography? It is a stationary phase, mobile phase, sample, capillary tube, chromatographic tank, scale, pencil. Stationary phase is the phase where sample will be applied. Mobile phase will be used to separate the components. Sample which is used to be separated or identified or analyzed or quantified. Capillary tube will be used to apply the sample on the stationary phase, and chromatographic tank will be used to develop the stationary phase. Scale will be used for the particular markings or to calculate the RF value, and pencil definitely to mark the markings it will be used. So firstly, to perform the chromatography, you need to cut your stationary phase, that could be TLC or the paper chromatography, into particular dimensions, 20 into 20, 10 into 5, 20 into 5, whatever the dimension we want or is the requirement of the practical. Then after marking the baseline, you will apply the sample on the stationary phase with the help of the capillary tube. You will add, put your selected mobile phase in the chromatographic tank, and you will allow the chromatographic tank to saturate by covering it with its lid. After saturation of the chromatographic tank, you will place your TLC or the paper chromatography in that chromatographic tank in such a way that your component or your spot of the sample did not get dissolved into the mobile phase. Cover your chromatographic tank with the lid and allow the sample to separate based on affinity. And when the chromatogram is developed, remove it from the chromatographic tank, allow it to dry. After the drying, calculate the RF values. This is the visual representation. Firstly, you have this is your stationary phase. This is your baseline. This black dot is the sample which was applied with the help of the capillary tube. This big jar is your chromatographic tank. This blue is the solvent which was used as a mobile phase. This is the lid of this chromatographic tank. When you applied this sample on the stationary phase, you poured your mobile phase in this chromatographic tank, covered it with lid, and allowed this chromatographic tank to saturate. Until that point, this spot will get dry. Then you placed your this chromatographic plate into this tank in such a way that this spot did not get dissolved in this solvent, as you can see in this picture. Allowed this stationary phase to develop for some time. Now you can see here, you can only say this is a black spot, but after development of the chromatogram, you can see three different spots. So after the development of chromatogram, you remove the chromatograph, and then you let it dry, and after drying, calculate the RF value. If I talk about this blue point or blue ink or blue spot, I can say this spot has more affinity for the stationary phase because it traveled very very small distance, while this spot has the highest affinity for the mobile phase. Those spots which will have the less affinity for the stationary phase, they will move faster, and those which have the higher affinity for the stationary phase, they will travel less distance. So this is a visual representation. Another visual representation. In this diagram, this jar is your chromatographic tank, and in this beaker is your chromatographic tank covered with the um, uh, covered with any petri plate or any watch glass. So, purpose of showing this diagram is chromatographic tanks can differentiate. I'm moving my cursor, and you can see this TLC plate is wet to this point. So this is the solvent front. This is the point where you decided you will let your chromatogram to develop. So this will be your solvent front. When you perform the chromatography, you let your chromatogram to dry. Then you have to find out the RF values. So this was your baseline. This was the point where you applied your sample. This was the solvent front. After separation, you remove this chromatogram, you remove this plate, you allow it to dry, and now you have to calculate the RF value. For this, you will calculate with the help of the scale the distance between the baseline and the solvent front, then the distance between the baselines to the spot, whatever the spot is. For example, this is green, this is a long spot, and then you have to draw a line at the center of that particular spot. This will be the distance of the green spot, blue spot, and the red spot, and you will divide these points with the distance traveled by the solvent front, and that value will be your RF value. So RF value must be less than 1. So in today's lecture, I told you about what is chromatography, its important terminologies, how RF value can be calculated, and in next lecture, I will be discussing with you all what are the principles involved in chromatography. Like if a component has more affinity towards the stationary phase, what does it mean? What are the basic principles due to which the components are separated, and which separation has the highest resolution? These points will be discussed in the next lecture. So I am open to your suggestions, uh, and I hope so. I delivered good content with you, and hope for the best. Thank you so much.